Today, since it's been a week, I think we'll actually just kind of review what we've covered and then move on from there. So we'll just do a real quick review. Okay, so good morning. I hope everyone had a good weekend. I am just now back on my feet. I'm glad to be ready for Monday. We are in chapter 18. We finished chapter 16. We finished chapter 17. We had an exam. There was a wide spread on the first exam, and that's pretty typical. The most common comment I usually get is the pacing of the questions. That is because this is supposed to be a closed book test and closed notes. And so we try and simulate that as best as we can. And part of that is by trying to not allow you enough time to Google or search through your notes or the book, things like that. I will be aware and try and keep the answer choices a little bit shorter on this next exam so you don't have quite as many to go through. But you kind of have a feel now for what it's going to be. And don't spend too much time on any one question, of course. And I know that it gets better. It does. Okay. This chapter 18 is on blood vessels, but most of the next exam, I think it's 35 questions. I'll have to figure it out a little bit later. But roughly come from the respiratory system. So I think about only 15 questions come from this one. I'll have to double check that, but it's definitely a disproportionate number. We started by talking about arteries and veins, about their tunics, about how there are different types of arteries. There are these large elastic arteries that capture that kinetic energy as elastic energy and then are going to release it for continuous flow. It's going to be places like the aorta. Again, they're called pressure reservoir. Their function is continuous blood flow. They have the most elastin found in any of the three types of arteries. And then we have these muscular arteries. They have a very thick muscular layer. They are active somewhat in vasoconstriction. They are less distensible, and these are going to be your named arteries. Arterioles are the smallest. They vary greatly. Some have 3 tunics, some have 1 tunic. These are where we're going to pick up total peripheral resistance because they are going to control the flow into and out of capillary beds. And today we may or may not get to intrinsic and extrinsic control of blood flow. We'll have to see. For capillaries, this is where we get our exchange of gas and nutrients. There are 3 types of capillaries: continuous, most abundant, least permeable. And then we get to fenestrated, which are going to allow for increased permeability at places like the kidneys, where we need to have active filtration and produce filtrate. And then we have our least common and our most leaky, so leaky that the blood flow becomes sluggish, allowing time for modification of large molecules. This occurs in the liver. You need to get reticulocytes into erythrocytes in the bone marrow, kind of push them out, as well as to seed platelets, and some in the spleen and some in the adrenal medulla. Capillary beds can be truly open. They can have a vascular shunt, which allows blood to flow, but not the exchange of gas and nutrients, or they can be completely closed if the arteriole will close it off completely. And then this would be all white and that there's no blood flowing through. So completely open, completely closed, or a vascular shunt where blood flows, but nutrients and gases don't exchange. Then we went on to veins, and we have capillaries drain into venules, which are these smaller, thin-walled vessels. They are stabilized by these pericytes, these smooth muscle-like cells, and venules give way to veins. Again, all 3 tunics, but a much smaller or much thinner tunica media, and is still a pretty extensive tunica externic because we need to have those collagen fibers and elastic networks to anchor veins, but also to stabilize lymph vessels and nerves. They're called capacitance vessels because they serve as blood reservoirs with up to about 65% of the blood volume located in veins. This is a nice pretty picture of a complete circuit. The heart is the pump. Then we have our arterial system, capillaries, veins. Anastomoses are just additional interconnections of blood supply. They're common in joints where mobility impedes blood movement, as well as abdominal organs, brain, and heart. And again, venous anastomoses are so abundant that you can lose veins, remove veins, and blood flow still occurs. Blood flow return. Okay, so this is kind of where we really start to get back to where we were. We're going to talk about volume. We're going to talk about force. We're going to talk about systolic and diastolic blood pressures. Blood flow is a rate. It's a volume per given period. The blood flow of the circulatory system is called cardiac output, and it's also usually expressed in either mils per minute or liters per minute. Blood pressure is force per unit area. We're going to talk a little bit about that. I'll come back. For blood pressure, we have systolic pressure is the highest pressure. It's when that is generated. And this would be at the level of the aorta. And then diastolic is when the heart is at rest. So this is the lowest pressure in the aorta. So the difference between systolic and diastolic is called pulse pressure. So pulse pressure is equal to systolic minus diastolic. And then mean pressure. First, I thought this was a typo until I realized it was referring to this darker blue line. Mean pressure generated by the heart as a pump is not an average between systolic and diastolic. It is going to be the mean arterial pressure is equal to diastolic plus 1/3 pulse pressure, because the heart spends more time in diastole than systole. So if it were just an average, then the mean pressure would be 100. But because of that additional time spent in diastole, it drops slightly below. And so for this example, it would be 93. If you are given a graph, you should be able to tell me systolic, diastolic, pulse pressure, and mean pressure. Probably not all at the same time. Those will be different questions, or it could just be one or two of those, but you will most likely see a graph that's representing pressure. The greatest drop off in pressure is at the level of the arterioles, and that's because total peripheral resistance starts to pick up. The pulse pressure starts to die off in the aorta and the large arteries, just as that elasticity it will start to dampen this difference. But the largest decrease in mean pressure occurs at the level of arterioles because of that increased total peripheral resistance. So resistance is opposition to flow. So blood flow is directly related to blood pressure or force. So the greater the pressure gradient, the greater the blood flow, but it's inversely related to resistance. So as resistance increases, then blood flow will decrease. Okay, and there are three factors that affect total peripheral resistance. One is blood viscosity, and this one doesn't change tremendously under normal circumstances, but increased viscosity equals an increase in resistance. And then these are the two that, well, blood vessel diameter is really the one that we're going to change. Total blood vessel length becomes important when we get to arterioles, And that's what there's so many, the length of the vessels is comprehensive. If you added all the arterial lengths and the capillary lengths together, it's quite significant. Okay. So for total blood vessel length, the longer the vessel, the greater the resistance encountered. And then blood vessel diameter is going to have the greatest influence on resistance. And that's why we are going to look at it again, how to adjust the blood vessel diameter. The reason why blood vessel diameter has such an influence on resistance relates to turbulent and laminar flow. Along the walls, the flow is turbulent and slow. And in the middle, we have laminar flow, which is smooth and fast. So if you have a larger diameter, a larger lumen, you'll have more laminar fast flow. If you vasoconstrict, you close down that lumen and you limit the laminar flow and you get more turbulent flow. You also will probably see a question that will give you a choice of vessels, and you'll need to choose the vessel. So it could be long and thin, it could be short and thin, it could be long and wide or short and wide. You'll have a choice of which one would give you the fastest flow and which one would give you the slowest flow. The larger the lumen, the faster the flow, and also the shorter vessel will be faster than this longer vessel. So this would be your best option for fast flow, and this would be your best option for low flow, but you'll have some other choices to pick out too. So if you understand the concepts, then you'll be able to pick the best one. So this was the relationship that I was discussing. That flow is directly proportional to the blood pressure gradient. It's inversely proportional to resistance. Arterial blood pressure is determined by elasticity as well as volume. We talked about how systolic is the peak pressure exerted during ventricular contraction. Diastolic is the lowest. Pulse pressure is the difference between systolic and diastolic. You'll need to be able to calculate or determine these from a graph, or if I give you like the systolic and diastolic, then you would need to be able to calculate pulse pressure as well as mean arterial pressure. So it will either be a combination of graphs and or where I give you numbers and you calculate it out. Capillaries are a low pressure system, and this is good because they're very thin. They can easily be damaged. Venous blood pressure, so the veins are also a low pressure system, and they have two adaptations that maximize like resistance to flow, and those are going to be that they have a large lumen, so they're going to have a lot of laminar flow, and then they have veins. So a large lumen and valves are their adaptations, and then systemic assists are the muscular pump from skeletal muscle, the respiratory pump from breathing, and then sympathetic venoconstriction. So the sympathetic system can get the smooth muscle to constrict and help with venous return. This becomes important also during exercise. I think we are back to where we were on last week. Regulation of blood pressure. There are three factors that are going to regulate blood pressure: cardiac output, peripheral resistance, and blood volume. If you substitute and rearrange, so if you substitute cardiac output, so the flow of the circulatory system for F, And then you rearrange this relationship so that to get rid of, I'm teaching it. It's funny, I'm reviewing this with one of my sons. If you need to get rid of something that's divided, you multiply by both sides. So change in pressure, so that mean arterial pressure, is directly related to both cardiac output and resistance. So this resistance that we're referring to is total peripheral resistance. And then cardiac output is something by something. There are two variables that go into cardiac output. Does anyone remember them? Heart rate times stroke volume. Okay. Mean arterial pressure is directly proportional to changes in heart rate, changes in stroke volume, and changes in total peripheral resistance. So you increase heart rate, you're going to increase mean arterial pressure, increase stroke volume, same, and increase total peripheral resistance. You'll also get an increase in mean arterial pressure. This one, you do need to remember that pressure is directly proportional to cardiac output and resistance. So again, this is rearranging and then just expanding, substituting pressure gradient for mean arterial pressure, because where we're going with this section is regulation of blood pressure. So how do we change this number? How do we increase it or how do we decrease it? Because chronic hypertension is going to damage our vessels and damage our heart, as well as other small vessels like in the eye and in the brain. And then a low blood pressure causes problems with perfusion of organs. So we need to keep blood pressure under a pretty constant or regulated manner. Anything that increases stroke volume, heart rate, or total peripheral resistance also increases mean arterial pressure. Stroke volume is going to be affected by venous return. Heart rate, we'll talk about the medullary centers like baroreceptors, and total peripheral resistance is affected by vessel diameter, vasoconstriction and vasodilation. And that's going to be under a couple of control mechanisms. So this is a good graphic. We're going to focus on how to increase or decrease mean arterial pressure. I added this in because it wasn't in there, but if you increase blood volume, you're going to increase our pressure, mean arterial pressure as well. But that's not really the focus of this section. So the focus of the section is cardiac output and total peripheral resistance. And if you increase stroke volume, increase heart rate, you'll increase mean arterial pressure. And then if you increase total peripheral resistance by vasoconstriction, blood viscosity, and then or blood vessel length. For regulating total peripheral resistance and cardiac output, there are short-term regulation via neural and hormonal controls, and then we can regulate blood volume to affect total peripheral resistance by long-term renal controls. So there are neural controls for total peripheral resistance as well as cardiac output. and then we'll get to the hormonal controls. Short-term regulation is acute. These are going to control, we're going to talk about short-term neuroregulation of blood vessels, so of peripheral resistance. We can alter blood vessel diameter, so decreased diameter increases resistance, and also alter blood distribution. These are under what are called vasomotor control. So there are vasomotor neurons that control vasomotor neurons are going to control that blood vessel diameter. So that is the neural control, is affecting the input to the rate of, okay, so we have blood vessels. We have a vasomotor neuron that is going to innervate these blood vessels. It normally has a middle level of action potentials of impulses, telling this vessel to maintain at a medium level. So they're not vasodilated, they're not vasoconstricted. These controls here are going to affect the rate of firing. So if baroreceptors get stimulated, or inhibited, depends, we'll go into that reflex arc in a second, they can increase the rate of firing, which would cause vasoconstriction, or decrease the amount of firing, so there's much less, and we'll get vasodilation. So the physical control is through these vasomotor neurons. They're the ones that are actually sending the action potentials down to the blood vessels and telling them to relax or constrict more. But it's these four neural control centers or receptors that provide the input to the vasomotor neurons on how they should regulate activity. So we'll look at these centers and how they affect the vasomotor fibers. So the fibers that control blood vessels are called vasomotor fibers, and they are part of the sympathetic nervous system. So vasomotor fibers cause constriction of arterioles at a moderate level, and this is called vasomotor tone. Then their activity, whether or not they're going to send more action potentials or less action potentials, are altered by baroreceptors, chemoreceptors, and higher brain centers. But it's these vasomotor fibers of the sympathetic nervous system that can cause the constriction of arterioles. So baroreceptors are going to be one of the centers that affects the rate of vasomotor firing. These are pressure-sensitive mechanoreceptors that are located in the carotid sinuses, the aortic arch, the large arteries of the neck, such as the carotid artery. So that's why if you go here to feel a pulse, it will actually start to slow your heart rate down. It's not the best place usually to feel for a pulse, but sometimes it's the only place that's accessible. So when there is an increase in blood pressure, then the baroreceptors, which sit in these muscular walls, and elastic, they are going to, that pressure expands them, and they actually widen, like they lengthen. And when they get stretched, then they start to signal. So increased blood pressure stimulates these baroreceptors. It stretches them, and it's going to cause them to increase their rate of action potentials to the cardioaccelerator and the cardioinhibitory centers. When baroreceptors are stimulated, they inhibit the cardioacceleratory center and stimulates the cardioinhibitory. So the result is a decrease in blood pressure. So when baroreceptors are stimulated, they increase the rate of firing to these centers that are in the medulla oblongata. Then the medulla oblongata is going to tell the vasomotor fibers to decrease their firing rate and vasodilate and decrease blood pressure. This is a really good graphic again. So we went this direction first, that blood pressure is increasing. These baroreceptors, they are mechanoreceptors. They actually get, they stretch and get deformed. That increases their input to these centers in the brain. Then the brain is going to send out messages it inhibits the vasomotor, so there's less stimulus to these blood vessels, and they will vasodilate. Decreased, so this is at the same time. So we have those short-term neurocontrols of total peripheral resistance and cardiac output. So short-term neurocontrols affect both. So when these baroreceptors get stimulated and signal the brain then decreases firing through the vasomotor, and then it sends input, it decreases the input to the heart, causing a decrease in heart rate, a decrease in contractility, and therefore a decrease in cardiac output. So baroreceptors can affect both total peripheral resistance and cardiac output, and it's a double, kind of a double booster to decreasing blood pressure. If blood pressure falls, these are not getting stimulated, they're actually inhibited. There's no input or less input to the brain. That lower rate of input then is going to stimulate vasomotor fibers, causing them to vasoconstrict, as well as increasing sympathetic activity to the heart to increase heart rate and contractility, and therefore cardiac output. So baroreceptors affect both total peripheral resistance and cardiac output as a short-term neural mechanism. There are also chemoreceptors located in the aortic arch, and again, some of the larger neck arteries, that they can detect increases in carbon dioxide or a drop in pH, so an increase in carbon dioxide, an increase in hydrogen ions, or a decrease in oxygen. These are going to then signal that cardioaccelerator centering to increase cardiac output, as well as increasing that vasomotor to increase vasoconstriction to increase blood pressure. What this increase in blood pressure does is it increases the perfusion rate, but it also affects the perfusion pressure at the lungs. So these are the two that I'm really going to go. So you need to understand how vasomotor fibers, if they increase their action potential rate, if you get vasoconstriction or vasodilation, baroreceptors and chemoreceptors. So baroreceptors, this negative homeostasis balance, chemoreceptors, that if they decrease, an increase in carbon dioxide or hydrogen ions or decrease in oxygen will increase blood pressure. And then these, I'm not really going to, these two would not be on the exam. They're kind of, I don't know if you remember from 1141, but we're always like, oh yeah, and the hypothalamus and the cerebral cortex, they also come into play. And they do. They have additional centers, but we're not going to go into those. What you needed to cover, you should have covered in 1141. The 3 topics are how vasomotor fibers affect blood vessel vasoconstriction or vasodilation, how the baroreceptor reflexes work, and then how the chemoreceptor reflexes work. So we talked about short-term neural controls, and now we're going to go to short-term hormonal controls. Short-term hormones result in changes in peripheral resistance, or we'll talk about long-term when we get to blood volume. When we increase sympathetic nervous activity, we can also get an increase in epinephrine and norepinephrine release from the adrenal medulla. This increases cardiac output and increases vasoconstriction. So if you increase the amount of epinephrine or norepinephrine, you're going to get vasoconstriction. We'll talk about angiotensin II in just a second. ADH, which is made in the hypothalamus, secreted by the posterior pituitary. You guys are having flashbacks to now enter the endocrine system, are going to increase blood volume through water reabsorption in those kidney tubules, and also in very high levels will cause vasoconstriction. These top 3, angiotensin II, which we will go into much greater detail, these all increase blood pressure. So epinephrine, norepinephrine, angiotensin II, and ADH all increase. These increase TPR and therefore increase blood pressure. Okay, so you have 1, 2, 3 factors that are hormones that can increase blood pressure. Well, the heart has its own protective mechanism. It says, yes, brain, yes, skeletal muscle, yes, abdominal organs, I understand that you need blood, but the heart has to have some sort of control mechanism to protect itself as well. So the heart produces atrial natriuretic peptide, which is going to antagonize aldosterone, decreasing blood volume and decreasing blood pressure. So we have 3 that increase blood pressure. We have 1 produced by the heart to protect itself, and it will decrease blood pressure. So for these 3, you just need to know if they increase blood pressure or decrease blood pressure. This is a table. It's okay. I think the best part of this table is whether or not it is going to increase or decrease blood pressure. So these all increase, and then we have atrial natriuretic peptide produced by the heart, which is going to decrease. I'm not as worried about the next two columns. So we covered short-term neural and hormonal regulation of total peripheral resistance and cardiac output. We didn't cover cardiac output as much because that was actually in 16 and 17. And now we are going to look at long-term regulation via the kidneys. So baroreceptors, which are the main input short-term, and this is, so like when we stand up, we don't faint, okay? But they are going to adapt to chronic high or low blood pressure. So they are ineffective for long-term regulation. That's kind of an important concept, is that baroreceptors are great in the short term, but under chronic conditions, they quickly reset their expectations. So for long-term mechanism, that is going to be done by the kidneys. We all go, why would the kidneys? Like, why pick the kidneys? Well, the kidneys receive 25% of your cardiac output at rest. Okay, that's a lot. And so it has a pretty good hold on what's going on in that circulatory system. So the kidneys, while you might not think they would be the pressure regulators, they're actually very well suited to it because of their direct relationship with cardiac output. So the kidneys rely on kind of a consistent pressure in order to produce glomerular filtrate. And then that glomerular filtrate goes into the tubule system. Some things get reabsorbed, some things get secreted, and we'll go into that with the urinary system. But we have an afferent arteriole and an efferent arteriole, and the pressure, these are leaky capillaries, the pressure of the system pushes filtrate into this capsule. By having a direct relationship between pressure and filtrate formation, that's going to allow for this direct renal regulation. So the kidneys regulate arterial blood pressure both directly, meaning they don't really have any control over it. It's going to be in direct response to the blood pressure, as well as indirect, which is where we get hormones and enzymes. The direct renal mechanism is really quite straightforward. When we have that glomerulus, like I said, it has an afferent arteriole, then it has glomerular capillaries, and it has an efferent arteriole. The amount of filtrate produced to go into this kidney system is directly proportional to the blood pressure. So if you increase blood pressure, you will increase filtrate formation, and then you will lose, you'll exceed the ability of these tubules to reabsorb, and you'll increase urine output. When you lose fluid, you're going to lose blood volume, and that will cause a decrease in blood pressure. So increased blood pressure increases the rate of filtration. The kidneys can't process it fast enough. More urine is produced, reducing blood volume and decreasing blood pressure. So that's pretty straightforward. This diagram on the left is going to show that when there's a drop in arterial pressure, then there is less filtrate produced, less urine formed, and that's going to increase blood volume in order to increase arterial pressure back up to normal. The reason why it's going to do a decrease in arterial pressure is to mirror what's happening on the indirect. but this one here at the bottom is probably the more common occurrence of how direct renal regulation occurs. But it wanted to be consistent with the next one. So direct has this relationship with filtration and urine production and blood volume. Pretty straightforward. The indirect mechanism for renal control of blood pressure is called the renin-angiotensin-aldosterone, either system or mechanism. We'll see it both places. And it looks kind of a mess, but it's not so bad. Okay, so we start with, it's kind of in two phases. This first phase is producing angiotensin II. So that's what we want to do first. So if there's a decrease in our arterial pressure, then we go through a cascade to get renin released from the kidneys. So a decrease in pressure increases renin. Renin is an enzyme. So renin catalyzes the conversion of angiotensinogen into angiotensin I. Angiotensin I is in the circulatory system, and there is an enzyme in your lungs called ACE. ACE converts it to angiotensin II. Do you need to know the whole cascade? No. What you need to know is a decrease in pressure stimulates the kidneys to increase renin production. When you increase renin, there is a cascade of reactions that results in an increase in angiotensin II. Then the second phase is that there are 4 functions of angiotensin II. Angiotensin II can act on the adrenal cortex to secrete aldosterone. If you remember, again, endocrinology, coming back to haunt you. Aldosterone is a mineral corticoid, and it is going to increase the sodium reabsorption by the kidneys, and where sodium goes, water goes. Okay, so that's where this water is here. So you also get water reabsorbed along with the sodium. That's going to increase blood volume and increase arterial pressure. This effect is a little less strong. So this definitely happens a lot, but it causes the posterior pituitary to secrete more ADH, which also increases water reabsorption by the kidneys. Okay, so it affects the number of water channels that are available, and it's going to increase water reabsorption. It also stimulates a thirst reflex or stimulus through the hypothalamus for increased water intake. And then it also acts as a vasoconstrictor. It's a hormonal control for vasoconstriction. Remember where we started? Blood pressure falls. Okay. I am in a car accident. I get a deep cut. I lose a lot of blood. My arterial pressure is decreasing. Then that is going to cause the kidneys to release renin. Renin is an enzyme, and it is going to, through a series of reactions, cause an increase in angiotensin II. angiotensin II acts in 4 ways to stabilize extracellular fluid and blood pressure. It's going to stimulate aldosterone secretion from the adrenal cortex. And then this is going to cause the kidneys to reabsorb sodium, which then water follows. So water is going to get reabsorbed also. It's going to cause ADH release from the posterior pituitary, which is going to cause an additional reabsorption of water by affecting the number of water channels. Triggers your hypothalamus to tell you to drink more. And then it acts as a potent vasoconstrictor, directly increasing blood pressure. Okay, so you need the first phase, decrease in mean arterial pressure, increases renin, increases angiotensin II. That's kind of one question. And then the 4 ways by which angiotensin II is going to stabilize blood pressure and affect the extracellular fluid. So the first 3 stabilize this extracellular fluid, and then this 4th one acts directly to increase vasoconstriction.
In this video, we will discuss the renin-angiotensin-aldosterone system. And before getting into all the complex stuff, let's have an overview of this system. So whenever your blood pressure falls, known as hypotension, the kidneys are able to sense it and they activate the renin-angiotensin-aldosterone system, which acts on kidney as well as the blood vessels to restore the blood pressure to the normal. Most of you know the kidneys are supplied by a set of renal arteries, which supply almost 20 to 25% of whole cardiac output to kidneys. This blood reaches the glomeruli through the afferent arterioles and leaves the glomeruli through the efferent arterioles. These cells you see are the juxtaglomerular cells. The word juxta means near, hence these cells are located near the glomerulus. These cells secrete a substance known as prorenin, and hypotension is a main stimulator of converting prorenin into renin. Renin is an enzyme which acts on angiotensinogen. The word angio means vessels. The word tensinogen means a substance which constricts the vessels. It is a plasma protein which is synthesized in the liver. Renin converts angiotensinogen into angiotensin I. The next step takes place in the lungs when the enzyme angiotensin converting enzyme acts on angiotensin I and converts it into angiotensin II. The angiotensin II is a powerful vasoconstrictor which mediates the effects of renin-angiotensin-aldosterone system. Let's look at the effects of angiotensin II on blood vessels. As I said, angiotensin II is a very potent vasoconstrictor. It constricts both the arteries as well as the veins. The constriction of the arteries leads to a total peripheral resistance increase, and the constriction of veins lead to increase in the venous return to the heart. Both these mechanisms increase the blood pressure of the person. This effect is rapid and takes a few minutes. Now let's look at the effects of angiotensin II on the kidneys. These effects are slow and take a few days to have their maximum effect. Angiotensin II acts on kidney in 2 ways. The first is the direct mechanism. As most of you know, the GFR of the kidney is directly proportional to the blood flow through the kidneys. The blood enters the glomeruli through the afferent arterioles, and angiotensin II, being a powerful vasoconstrictor, constricts these afferent arterioles mainly and decreases the blood flow through the kidneys. This results in a decrease in the GFR of the kidney, which results in a decrease in the urine output through the kidney. Now, second is the indirect effect, in which the angiotensin II acts on the adrenal gland and releases aldosterone. Aldosterone in turn acts on the kidney and increases the sodium reabsorption as well as the water reabsorption. So we have a decreased urine output, increase in the water reabsorption, and increase in the sodium reabsorption. All these effects lead to an increase in the extracellular fluid of the body, an increase in the capillary pressure, and a long-term increase in the arterial pressure. So to summarize, let's suppose a person gets stabbed and he loses a lot of blood. This will lead to a decrease in the blood pressure of the person known as hypotension, which is a potent trigger of activation of renin-angiotensin-aldosterone system. The RAAS further acts on the blood vessels as well as the kidneys and normalizes the blood pressure. So this was all about the renin-angiotensin-aldosterone system. Thank you so much for watching.
All right, we are back to us. Okay, so this is, I think, actually a little bit of a complex diagram. It's a little busy, but it does provide a summary of the effectors on blood pressure. So on how to, and it's, to me, it's better if you start at the bottom and you look at increasing mean arterial pressure can be accomplished by increasing cardiac output or increasing total peripheral resistance. The three factors that go into total peripheral resistance are where we kind of started today with diameter, blood viscosity, and blood vessel length. And then blood vessel length is affected by body size. Dehydration or living at altitude will affect blood viscosity. Vasomotor tone, I've probably said that 100 times, as well as on Wednesday, we'll look at the chemical factors. Today, we only looked at the neural factors of vasomotor tone on blood vessel diameter. And then if you see is going to affect both pathways. This is that short-term neuroregulation through baroreceptors and chemoreceptors affecting both cardiac output and total peripheral resistance. And then we had the renin-aldosterone system, and that is going to lead to the increase of sodium and water affecting extracellular fluid, and as well as venous return to increase mean arterial pressure. So kind of a complicated slide, but if you go from the bottom and you work through each pathway, it makes a lot more sense. We've got 3 slides left to finish off 18.8. It's just hypertension and hypotension. So hypertension is an elevated, and it's usually, we're always worried about chronic hypertension. We're usually not so worried about transient hypertension. But the value is 140/90. It can be transient during fever, getting sick, being upset. But then what we're really concerned with is prolonged hypertension, because prolonged hypertension is one of the major causes of heart failure, vascular disease, renal failure, and stroke. It can be defined as primary or secondary. And I don't like these terms, but I'll go with them because that's what people use. So 90% says no underlying cause identified, which I find is like the most hilarious statement ever, because we do know the cause. So these are because, well, there's heredity, but there's diet. This high salt intake is going to increase your extracellular fluid, and it's going to cause hypertension. Smoking is under there too, and we know that nicotine is a vasoconstrictor. So again, I'm not quite sure why we say no underlying cause. But primary hypertension is by far the most common, 90%, and it is due to what they call risk factors, which I would call causes, but no textbook will say that, so it's just me. But these risk factors are diet, especially a high sodium diet, obesity, because this increases the amount of blood vessels that you have. Age is not surprising. Our sympathetic nervous system really starts to decline with age. So Anything that such as vasomotor, which is under total control of the sympathetic nervous system, no parasympathetic input at all. It's really not surprising. So this is really due to a decrease in regulation of our sympathetic nervous system. Diabetes mellitus, stress, and smoking. Nicotine is a vasoconstrictor. No cure, and I kind of disagree with this too, because if you correct these factors back to what are considered normal values, many people they're no longer hypertensive. Okay, but if they are, then there are antihypertensive drugs. Secondary hypertension is less common, and it says it's due to identifiable disorders, such as problems with the renal arteries, kidney disease, endocrine disorders. So these are focused on correcting the underlying cause. Usually in this class, I will say we kind of tend to focus more on this one, because these are things that we can, well, one, if you are working with probably anyone over the age of 40 or 50, you may be involved with administration of antihypertensive drugs. So know the risk factors, know that this is the most common. Okay. Hypotension is considered 90 or below 90/60. Usually not a concern. People who tend to have low blood pressure, they adapt to it. Orthostatic hypotension is temporary low blood pressure, possibly dizziness when suddenly rising. Again, this is more of an issue with people as they get older, because their sympathetic nervous system doesn't compensate as quickly or as completely. Chronic hypotension, sometimes associated with hypothyroidism. And then acute hypotension can be due to circulatory shock. But the main ones are the value and what orthostatic hypotension is defined as. Okay, so I think, yep. So this is a good place to stop, because control of blood flow is it's not nearly as long as 18.8 to cover, but it certainly can't be covered in the next 5 minutes. So we will just stop here and pick up on Wednesday.